This is information you need to review to answer the P-Glow transformation quiz before we do the transformation on June 23rd. So bacterial transformation is where you get a bacteria to take up some extra DNA. And in this case, the DNA that it's going to take up is known as a plasmid. Green fluorescent protein is a protein that was discovered in the 1960s, and this protein glows green under ultraviolet light, and that's because it has a special chromophore in the middle of it. So this is the structure of it. It's a beta barrel, meaning it's got mostly beta pleated sheets, and it's actually hollow with a chromophore in the middle. In the 1980s, the gene was cloned and it was inserted into a plasmid. So the plasmid is the vector. And so if you can get bacteria to take up this recombinant plasmid, you can express the protein in bacteria and see it glowing green under UV light. So it's a very cool experiment when it works. So let's start off with our EdvaTech P-Glow plasmid. This plasmid has a number of important parts. The first is a plasmid must have an origin of replication so it can replicate. The second, it has to have a multiple cloning site, and this is where the green fluorescent protein gene was actually inserted. In order to express this gene, you need to have a promoter, because remember, promoters switch on gene, and in this case, the promoter is known as a T7 promoter. And finally, we have to have a way of selecting which bacteria actually took up the plasmid, and that's an additional selectable marker, and in this case, it's a gene for ampicillin resistance. Ampicillin resistance is a gene that codes for a protein that breaks down ampicillin. So if our bacteria, E. coli, has taken up this plasmid, it will be able to grow on ampicillin. So here's a little bit of the details. So this is our host chromosome. The host chromosome has a lac promoter in it, which will switch on the T7 RNA polymerase gene. In order for this to be switched on, we're going to use our inducer known as IPTG. And IPG will remove the repressor that allows this gene to be transcribed. It's going to make host cell, so the host cell RNA polymerase is going to transcribe the T7 RNA polymerase gene, which will make messenger RNA and then be translated into a protein. This T7 RNA polymerase is now going to be able to switch on the green fluorescent gene. So the T7 RNA polymerase, once it's been translated into a protein, so the T7 RNA polymerase is a protein, it's going to bind to the T7 promoter on our plasmid, and the gene for GFP will be made into messenger RNA and then translated into green fluorescent protein inside the bacterial cell, and we'll be able to see it. So within the package you received before class, there's a set of instructions. I really encourage you to go through it, how we're actually going to make our E. coli take up this plasmid. So this is the quick version here, which is in your packet. And then the important thing to know is that we're going to plate out 4 plates when we're done. 2 are control plates and 2 are experimental plates. The plus plates have the plasmid DNA added to them, and the minus have no plasmid DNA. Therefore, the minus place will just be straight E. coli, so it should grow up as a complete lawn. If it has minus DNA plus AMP, then that E. coli should not be able to grow unless it's ampicillin resistant. And then our 2 experimental plates got the plasmid. So if the plasmid is there, it should be able to grow up on AMP. So on this plate, we should see some white colonies because the inducer is not there. And finally, on this last plate, plus AMP plus IPTG, we should be able to see our green fluorescent colonies. So read through the instructions, the detailed steps, we'll follow them in class. But what you need to do before you come to class is answer these pre-lab questions. These answers are basically in the handout that I've given you, as well as in the video that I have just done. And then make sure you answer the quiz questions on P-Glow before coming to class on June 23rd. And let's make our bacteria glow green. Should be pretty cool if it works. And it should work. It does work. It's worked in the past.